Hello everyone, this is Dr. Karen's, and today I'll be talking about Chapter 9 for Microbiology for Non-Science Majors at Eastfield. So this chapter is focused on different methods, chemicals, other ways to control microbial growth in the environment. So whether you're talking about a household surface like a kitchen counter or an operating room in the hospital, we're talking about environment, surfaces, things like that, not the human body, and that's for a different chapter. All right, so before we get started, we need to discuss a few terms, and these are listed in Table 9.1, and they're important because a lot of times we tend to use some of these terms interchangeably, but technically they mean different things. So first I want to start with a couple of suffixes. So there is this sidal suffix versus static or stasis suffix. So I'm just going to use the general term microbe to talk about these suffixes. So a microbicidal process or method or chemical is something that is going to kill and completely destroy a microbe. If you want to be more specific, you can say a bactericidal or bactericide or fungicide. And whatever prefix you're using is going to tell you what particular microbe that agent is working against. But that suffix cidal means kill, means complete destruction, versus the static or stasis suffix like microbiostatic, which means just to inhibit microbial growth, slow it down, but not completely kill microbes. And depending on what environment you're trying to control microbial growth in, a microbial static agent might be sufficient to keep people healthy, like in your home, versus in a hospital where you might want a microbiocidal agent because you have a lot of sick people with immunocompromised systems that can't handle any kind of germs. So you need to be more careful there. Some other terms: antisepsis versus disinfection. Both of these terms are referring to reducing the number of organisms or destroying some or most microbes, but the difference is where are you doing that destruction. If you're using a chemical that is going to reduce the number of microbes on living tissue, then that chemical is an antiseptic. But if you're using a chemical, it could be the same chemical, you're using it to destroy some or reduce the number of microbes on non-living tissue, like a kitchen counter, then that's a disinfectant. And so some agents are only antiseptics, some are only disinfectants, and some can be both. So for example, rubbing alcohol, isopropyl alcohol, you can use that as an antiseptic when you're cleaning a cut on your hand, or you can use it as a disinfectant when you're maybe trying to clean a bathroom surface or something like that. This term degerming, that is a mechanical method. So it's not a chemical. It's not something that actually either kills or reduces or inhibits the growth of microbes. It is mechanical, physical method of removing microbes. So the process of washing your hands, when you're physically rubbing your hands together, that is physically stripping microbes off of your skin. This is why, one of the reasons why when they say to wash your hands, especially during this pandemic time, why they're telling you to wash your hands for 20 seconds, because that's the time that was determined to properly de-germ your hands. You can de-germ your hands with just water. You don't have to have soap to de-germ them, but the soap does make it easier. It makes it easier to rub your hands together, but the soap will also grab onto some of those microbes, and we'll see why in a second. And if there's antimicrobial properties to that soap, that's going to help kill some of those microbes too. Pasteurization is it's a kind of like a way to disinfect beverages, and other types of food. It's specific for food. We'll talk about that in more detail in a minute. And then sanitation, again, it could refer to disinfection. It could refer to sterilization, which is destruction of all microorganisms on an object. But sanitation is a specific term you use when you're talking about public health standards. So like in a restaurant, at a school, things like that. And then again, sterilization, destruction of all microorganisms in or on an object. That's like the be-all, end-all method. And you might not need to technically sterilize your bathroom surface even. Like there are some environments that need to be sterilized and some that will be fine to just disinfect, and you still won't get sick. Okay. Another thing to think about when we're thinking about these different methods is microbial death rate. So this has to do basically with how long a microbe needs to be exposed to one of these methods to kill all of the microbial cells that are on that surface. Different microbes will have different death rates. So the example they show you in figure 9.1, I don't think they tell you what the microbe is and what's being used on it, but it's showing you that every minute that microbe is exposed to whatever method is being used on it, 90% of those microbial cells die. So if you're using some kind of cleaner, household cleaner on these, let's say it's a group of bacteria, if you expose those bacteria to that cleaner for 1 minute, 90% of the bacterial cells will be killed, but not all of them. And so after 2 minutes, 90% of what's left will be killed, and so on and so forth. And if you continue this line, where and when you get to no microbes left is about 9 minutes. So for this microbe, it takes about 9 minutes to kill all of it. This is an interesting thing for you to do. If you go and find a household cleaner, like a bathroom cleaner, something that you use in your home to disinfect a surface, so most likely a bathroom cleaner or a kitchen cleaner, go and find that and look at the instructions. Usually there are 2 sets of instructions. One for cleaning and one for disinfecting. And the disinfecting instructions are specific and they tell you, let's say it's some kind of kitchen counter antimicrobial cleaner. It'll tell you, spray your kitchen counter with this product and let it sit and keep that counter wet with that product for X number of minutes. And the reason for that is because they're averaging, whoever designed the product is averaging the death rate for the most common microbes that would be found in your kitchen. And they figured out, oh, it's going to take this many minutes of exposure to that product to kill everything that might be on that kitchen counter. And that's different from cleaning. Now, if you're physically rubbing that counter with that paper towel or whatever, that's some degerming that you're doing as well. So is it effective to just spray that disinfectant and wipe it right away? It might be effective, but it might not be as effective as if you were to follow the disinfecting instructions. So I just thought that was interesting because I don't think a lot of people are aware of that. And I know I don't always read the instructions on my cleaners, and it can be eye-opening when you do that, especially if you look at your bleach. I didn't know this until just recently, but there are different types of bleach that you can buy at the grocery store. They all look the same, but they don't all disinfect. Some just whiten. Others will actually disinfect. So if you're using bleach to clean something and disinfect that surface at home, you might want to go check and see if it really does disinfect. Okay, so we're going to be talking about some microbial control methods in a minute. But what exactly are these methods doing to the bacterial cells or to all these other microbes that it might be killing or inhibiting? And there's two main mechanisms specifically for bacteria. One is these methods will target cell walls of bacteria. So if you affect the integrity of that cell wall, either you weaken the cell wall, you poke holes in the cell wall or something, that cell is going to burst and therefore the cell is going to die. So targeting the cell wall is a great way to kill or inhibit the growth of bacteria. Another method is to target proteins or nucleic acids. If you target the proteins of bacteria, you're going to target their enzymes because those are proteins. And if the bacteria don't have working enzymes, they're not going to be able to do any type of metabolism and they're not going to survive. Same with nucleic acids, like RNA. If you can't make RNA, you're not going to be able to make proteins. You're going to die. So the two main targets for these methods that we're going to discuss in this chapter are the cell wall of bacteria and then proteins and nucleic acids. If you are a company that is trying to design some type of antimicrobial agent, there are things to consider. Like, yes, you want it to be able to kill things, but there are other things you need to think about as well, especially if you're thinking about a household cleaner. You want it to be inexpensive. You don't want to have to pay a lot for your bleach or whatever you're going to use. You want it to be fast acting. You don't want to have to spray your kitchen counter and then wait an hour for it to work. Stable during storage. You don't want to have to store your bleach in the refrigerator. And then the most important thing is, yes, you want it to control microbial growth, but you don't want it to harm you or the people around you or your pets, or you don't want it to harm the surface that you're using it on. So you want it to be safe for everything except the microbes. So you need to think about all of these things when you're designing one of these agents or even trying to pick one to use. You got to think about, well, where am I using it? What is the environment? And how strong of an agent do I actually need? This is figure 9.2. It is showing you, giving you a list of different groups and kinds of microbes and putting them in the order of how resistant they are to most of these antimicrobial methods. So at the bottom, you have enveloped viruses. Those are the most susceptible, meaning they're the weakest of these microbes and they're the easiest to kill. Whereas at the top are the things that are the hardest to kill, the most resistant to these methods. Prions, we haven't talked about that yet. We'll talk about those when we talk about viruses. They are the hardiest things to kill. Endospores, we talked a little bit about endospores already. Those are hard to kill because they can survive harsh conditions. So again, if you're trying to design some type of cleaner or disinfectant antimicrobial agent, you need to think about, well, what groups of microbes am I trying to target? And that will tell me how strong my agent has to be. Okay, let's move on to these methods. So we're going to talk about a few different groups of methods of controlling microbial growth in the environment. The first of these groups are physical methods, and the first of this group are heat-related methods. So high temperatures are going to denature proteins, affect enzymes, proteins in the cell wall of bacteria, in the membrane. It's going to disrupt a lot of things and affect growth and metabolism. But there's moist heat and there's dry heat. And they do different things. So moist heat would be things like boiling, pasteurization, something called autoclaving we'll get to in a second. Moist heat can disinfect surfaces, can sanitize, can sterilize. It's going to do all those things we talked about earlier with the proteins and membranes. Moist heat is more effective than dry heat because heat is conducted better in water than it is in the air. So these methods really quickly. Boiling. This is a kind of a low level germicide, meaning it's going to be able to kill the weakest of those microbes that we saw in that list. Boiling time is critical depending on what you're trying to kill and at what elevation you're at. The hardier things like endospores and stuff, those can survive boiling. Autoclaving. This is a method that is used in the laboratory setting and in hospitals. It's kind of like an autoclave I have a picture here. This is a very small autoclave. They're usually as big as like a refrigerator. It's like a giant pressure cooker, basically. So you're using steam, boiling water, pressure, really high temperatures to sterilize. Is a sterilization method. We use it in the lab. In the lab setting, we use metal inoculation loops. You can autoclave those. Any type of glassware that you use in the lab that has been contaminated with microbes, but you don't want to throw it away, you can throw it in the autoclave and it'll sterilize and kill it, and you can use everything again. Pasteurization. So I mentioned this before. This is a method of treating beverages and other foods to inhibit microbial growth but not affect the taste of those foods. So it's usually high heat for low amounts or short amounts of time. It does not sterilize your milk. You think of milk often when you think of pasteurization. It's not sterile. It's not completely free of microbes. You've just lowered the numbers enough that milk is going to be safe to drink. Now, once you open that milk, you've introduced it to microbes, and that's why your milk will eventually go bad. There are different types of pasteurization, but they all involve high heat. The more modern types, this is table 9.2, it's showing you these types of pasteurizations. The more modern types that are used often, I see it a lot with organic milk and other organic products. Really high heat, higher than boiling, for just a few seconds. And it makes, honestly, any milk, for example, or a dairy product that has been treated in this way is shelf stable. Even though you buy your milk in the refrigerator, in the refrigerated section, it doesn't need to be refrigerated until you open it. And you can even get some milk on the shelf. There are those little milk boxes, like juice boxes. Those are shelf stable. You can drink them at room temperature, but once you open them, they need to be refrigerated. Okay, so those are the moist heat methods. Now we're going to go to dry heat. You would use this method on anything that can't get wet, and you would use it for sterilization. It's not as effective as moist heat, so you need to expose your microbe to dry heat for longer amounts of time. But a great example of this is incineration. So when in the lab you have your metal inoculation loop, you sterilize it by running it through that Bunsen burner flame, and that is going to kill everything on that loop. Other physical methods of microbial control: refrigeration and freezing. When we think about these methods, we're like, well, that's what I use to keep my food in so it doesn't go bad. Yes, because those methods are inhibiting microbial growth. So both of these methods are going to be microbiostatic. They're not going to kill things. They're just going to slow the growth down. So refrigeration will work for a certain amount of time, but not forever. As you know, you have an open gallon of milk in there, it's going to go bad at some point. But it will hold off microbial growth, usually for long enough so you can eat and consume those products. Freezing is going to work for longer periods of time. Other physical methods often related to food are desiccation and lyophilization. So desiccation is drying, like dried fruits, dried grains, pastas, things like that. you completely remove, or you remove most of the water, and it's going to inhibit a lot of microbial growth that way. It's a way to preserve your food so it lasts longer. Lyophilization is freeze drying, and that can be more long term. There are some foods out there that you can buy that are freeze dried. Like there are freeze dried fruits out there. The consistency is more chalky than like a regular dried fruit. All right, another physical method of microbial control is osmotic pressure. This has to do with using sugars and salts or solutes to create hypertonic solutions around your bacterial cells so that water gets drawn out of those bacterial cells and you kill those cells or inhibit their growth. And we use this, again, in food preservation. You use sugar to preserve your fruits in the form of preserves. That's why they're called preserves. You can use salt to preserve your foods like beef jerky and other things that are salted and salty, but the salt is keeping microbial growth at bay. Another physical method is radiation. So there's ionizing radiation and non-ionizing radiation. What do they do? Ionizing radiation is kind of pulling electrons off of atoms inside bacterial cells, creating ions, and the ions are very disruptive to other molecules and things within the bacterial cells, and it's going to kill the microbe. Examples of this are gamma rays and X-rays, and gamma rays can be used to treat your produce. If you see this insignia, this little green circle with the flower in it on your produce, you know that your produce has been irradiated. It doesn't mean that it's radioactive. It doesn't mean that you're going to be exposed to the radiation. It just means that they've been irradiated to kill anything, any molds and things like that that might be growing on those bacteria or on those produce. Non-ionizing radiation doesn't strip off electrons, but it excites them and they form new bonds. So like in a protein inside a bacterial cell, if that protein is exposed to non-ionizing radiation, some of the bonds within that protein might change, and it will change the structure of that protein. Change the structure of the protein, you change how well the protein works. UV light does this. It specifically affects bonds within DNA. And that's why it can cause cancer in humans. But in a way, it can kind of cause cancer in bacteria too. You can use UV light actually to disinfect surfaces, disinfect the air. And in terms of that, it can be less damaging to the surface because you're not getting the surface wet or anything like that. But you do have to expose that surface, for, depending on the surface, could be maybe 20 minutes or so to that UV light. Table 9.4 summarizes all of these physical methods we just talked about. It specifically tells you what that method does to the microbe. That's under the action category or the action column. Does it destroy proteins? Does it affect the cell wall? What exactly does it do? Okay, so those are the physical methods. Now we're going to talk about chemicals, different chemicals you can use to control microbial growth in the environment. Similarly to those physical methods, these chemical methods are going to affect cell walls, cell membranes, proteins, or DNA. So let's just go through this laundry list. So phenols and phenolics. I think Lysol falls under this category. What do? Denature proteins, disrupt cell membranes. Often used in healthcare settings, sometimes in homes. They could have side effects, so it just depends on which particular phenol you're talking about. Alcohol, so rubbing alcohol. This is going to be intermediate level. It denatures proteins, disrupts membranes. You can use it as an antiseptic on yourself, on a person's skin. You can use it as a disinfectant on a surface. Halogens, so these are things like Iodine, chlorine, chlorine bleach, those types of things, they're going to denature enzymes. Iodine is still used in the healthcare setting. That's what this figure 9.14 is showing. It's that orangey color, that's iodine, and that can, in this case, it would be an antiseptic. You can use it as a disinfectant too. Oxidizing agents. So they're going to affect enzymes in a microbe. They can be a disinfectant, they can be antiseptic. Examples, hydrogen peroxide. It can disinfect and sterilize surfaces. And this I found interesting when I first started teaching this class. This is where I learned that you can't and shouldn't really use hydrogen peroxide on open wounds. So if you have like a cut on your hand, most people would turn to either rubbing alcohol or hydrogen peroxide. You would often find both in somebody's medicine cabinet to disinfect or to serve as an antiseptic for that wound. But hydrogen peroxide isn't useful against open wounds because catalase can inactivate it. It doesn't work. So I found that interesting. Ozone can be used to treat drinking water, and there are other chemicals here too, but they're basically going to affect microbial enzymes. Surfactants. So anything that is soapy and slippery like a soap is going to be a surfactant. They reduce the surface tension of solvents and they help kind of grip and trap microbes. They don't actually kill or affect microbial growth in any way. Instead, they're good for degerming. They help strip those microbes off your hands when you're washing your hands with soap. Heavy metals, heavy metal ions, like silver nitrate. There are other examples out there too. They're going to denature proteins. Low-level bacteriostatic agents can be used as antiseptic. They used to put silver nitrate on, or as a treatment for gonorrhea to prevent blindness. Copper can control growth of algae. Thimerosal, that's got mercury in it, that used to be used to preserve vaccines. I think they use other things now. Aldehydes, think formaldehyde. Formaldehyde is a preservation chemical. So if you're doing any kind of dissections, those specimens are usually preserved with formaldehyde. When we say preserved, we mean we're trying to prevent microbial growth so the specimen doesn't rot and go bad. So you use these types of things for I would say mostly in the hospital setting is when you would use it for disinfecting rooms and instruments and things like that. They're going to denature proteins and affect nucleic acids. Gases. Different gases can be used in a chamber to sterilize things. They could denature proteins. They can affect DNA. They're poisonous and explosive. So we don't use them a lot, and you definitely want to make sure there aren't people around when you're using them. But they're advantageous if you have something that you need to sterilize that you can't get wet or is really big and cumbersome that you can't disinfect or sterilize in any other way. So like a mattress, if you're going to donate a mattress somewhere for somebody else to use, ideally whatever company is taking it from you is going to disinfect it or sterilize it, and you can't get that wet. You can't incinerate it. If you want to give it to somebody else, you need this other way to sterilize it, and putting it in some kind of like a gas chamber could be a way of doing that. Enzymes. There are antimicrobial enzymes that can work against microbes. Your tears contain an enzyme called lysozyme that actually affects the cell wall of different bacteria. But there are other enzymes that can do those types of things too, that you can use on instruments, on surfaces, things like that. Drugs. So antimicrobial drugs, yes, these are chemicals, so it is a chemical method of microbial control, but this really is more relevant to Chapter 10 because Chapter 10 is talking about antimicrobial control in the body. So an antimicrobial drug, you wouldn't use that on a surface. You're going to use that in the body, and so that really belongs in Chapter 10, so I'm not going to talk about that now. Table 9.5 gives you a list and a summary of all of these chemical methods, including the action or what it does to the microbe. All right, so going back to this example of you trying to develop some type of cleaner, household disinfectant or whatnot. You have to think about, what surfaces you want to treat, who's going to be using it, what microbes you're trying to target when you're developing this project. But then you also want to know, does my product work? You don't want to send out your product before you know if it works or not. So how can we figure out if these disinfectants and antiseptics actually work? One way is, and it's not really a way that's used anymore, but it's called the phenol coefficient, where phenol used to be the standard. And so you would test your product to see how well it compared to phenol, basically. But we don't actually use that anymore. The United States uses this used dilution test. So what do? They have these metal cylinders. They dip it into cultures of bacteria. Then they dip it into different dilutions of the disinfectant that they're testing. And then they dip it in a tube of sterile media to see which dilutions of disinfectant still allow growth of that bacteria. And the idea is you want to find the most dilute of that disinfectant, the most diluted version of that disinfectant that completely inhibited growth. And that's the concentration you would go at when you develop your disinfectant for mass production. The Kelsey-Sykes test is something I think that's used in Europe. It's the same kind of thing. In this case, there's bacteria that's being added to different dilutions of your disinfectant you're trying to test. You remove the bacterial samples at different times and incubate them and see what grows and what doesn't. So you're looking for the most, or like the effective disinfectant dilution that you're looking for is the one that stops growth in the least amount of time. The in-use test, this is something where you can kind of test things immediately. So you swab objects, like maybe let's say a doorknob, before and after you apply a disinfectant that you want to test, and then you try to inoculate medium after you've applied the disinfectant, and you're looking to see if you get growth after you've applied that disinfectant or not. All right, we'll talk about resistance in the next chapter. I think that's pretty much it. So this really is more like a laundry list of different types of methods. And if you look at the study guide, what I'm asking you to know is, do you know what these methods are and how you do them? And how strong are they? What are they killing? What are they affecting? And what specifically are they doing to the cells? Are they affecting the cell wall? Are they affecting proteins? Are they affecting nucleic acids? What are they doing? So this would be a good opportunity for flashcards. If you've never done that before, when you have a chapter like this where you just have a list of things and a certain number of items that you need to know for each of those things in the list, this is a great opportunity for flashcards. So that's my recommendation for you.